Hello, good afternoon, ladies and gentlemen. So today we have a professor visiting from Sweden, actually a full professor at Uppsala University, and his name is Leif Hammarström, who is famous as photochemistry and photo-induced spectrum transfer and the proton transfer, and also the PCT area. He's a very famous professor. And before starting, asking him to start his lecture, I would like to spend some time to introduce his career so far. So he got I heard that he's for a long time staying in the same city and same university. And actually, he came with his wife, and his wife is also originally from this Uppsala city. And he got a PhD degree in 1995 in physical chemistry at the Uppsala under the supervisory of Professor Almgren. And then he went to Bologna to be involved in the Vincenzo Bausani group in Italy for doing postdoc for 1 year. And then he got a researcher position coming back to Uppsala in 1997. Then 1 year later he became assistant professor of the same university. In 2001 he became associate professor in physical chemistry at Uppsala University. And then in 2002 he became a research fellow of the Royal Swedish Academy of Science. It seemed to be a very high standard award type. At the same time, he said he got this position, this kind of award, and he got a 5-year research fellow position at the Royal Swedish Academy of Science. He told me. And he also has many awards, which is too many, so I cannot introduce all those things. And he's a leader now. He's a leader of the Swedish Artificial Photosynthesis community. So he's a leading professor in this area. And I think this is a story about him. So I would like to ask him to start his lecture on the molecular and biomimetic approaches to artificial photosynthesis. So, Leif, please start your lecture. Thank you for coming. So let's stand.
Thank you very much. Thank you, Ken Sakai, for being kindly hosting me during my stay here in Japan with JSPS fellow, and thank you for the kind introduction. So I know that many people here work on renewable energy and also solar energy, but also I heard from Ken that there are some people who don't, and several students. So we agreed that I will do a little bit of general introduction, general comments on artificial photosynthesis from an energy perspective. These are two pictures from Uppsala, my hometown, and this is the old cathedral. This is the main cathedral in Sweden. This was started to build in 1260, and this is a much newer building. This is the Hammarström laboratory where I work. So this is a center for the campus for mathematics, physics, chemistry, engineering sciences, and also here, that part that you don't see is information science and technology. We use a lot of energy in the world. In 2013, we used on the average power consumption of 18 terawatt, mostly fossil fuels of different kinds. And the predictions are hard to do, but many sources give the prediction that we're going to use about twice as much energy by 2050. And if we don't want to increase the use of fossil fuels, we have a large gap to fill. It's not easy to fill all this maybe 15 or 20 terawatt extra here. Why do we need to have 20 terawatts extra? Some people say, can we save energy? Just save the energy, don't use so much energy. But there's a problem here, And there is, first of all, 3 billion people on the planet are not born yet. We will be about 3 billion people more. And, and people are not yet born. They cannot save energy. So they definitely need energy. And if you look also in the energy use per capita, this is from Gapminder, a Swedish site that has become quite famous. This is the energy use versus GDP per capita, the economy of the country. And this is a logarithmic scales. And you can see that there's a color coding and a big dot. So a big dot means a country with many inhabitants. So this yellow one is probably United States. This big red one is probably China. And this big blue one is India, etc. And you probably find Japan at one of the red dots here on the top, with high income and high energy use per capita. So there's a very strong correlation here. And you see also that many countries here don't use a lot of energy, and they cannot save. And one reason we cannot save is also this. This is from Gapminder. This is a health, measured as life expectancy at birth. How many years on the average the newborn child is expected to live? From 45 years to 85 years. Again, versus GDP per capita. There's a much bigger scatter in this graph, but undoubtedly there's a correlation going something like this, with a high GDP meaning a long lifetime. And you can see many countries here in blue are in Africa, south of Sahara, for example. And you see Japan over here, unusually long lifetime. And so the use of energy means that you can have a high GDP, and a high GDP means that that your children have a chance to survive until they're grown up. That you can they can have education, you can have food on the table, etc. So we're not talking about luxury consumption. Maybe in Japan and Sweden and other countries we have luxury consumption, but in most of the world, it's a question of survival. So we're going to need a lot more energy in the future as the population grows, and these countries have a chance to grow, to have a better life. Compared to these 20 terawatts, the incoming solar power to our planet is huge, 120,000 terawatts. So in about 1 hour, it's equal to what we consume in a full year. And if we could just harvest 0.02% of that number, we would fill the hole that we need. You can do solar energy conversion in many ways, and a very old way is of course to do biomass. But biomass conversion is very inefficient. These are the most efficient, highest efficiency that people have recorded for biomass growth, and these are for a limited time. So it's not quite sustainable. These are for miscanthus and switchgrass, for example. This happens to be an example of Sweden, and here you can see that the percentage is much lower. But to produce an equivalent of about 10 terawatts of biomass, we have to grow biomass in all the land, agricultural land. So we have to stop eating. So if you want to solve the energy problem by biomass, we will not go all the way, and we'll have to stop eating. Can't we make photosynthesis more efficient? If you want to look into the efficiency losses of natural photosynthesis, this is a very good source, Annual Review of Plant Biology. Here they go through the energy losses for different types of plants with different metabolism. And you can see that these are theoretical limits, 4.6 and 6.0. This is theoretically the highest you can expect to get from a plant. And this could be compared, for example, with photovoltaics, where the single band gap semiconductor could give over 30% theoretically. If you don't make glucose, microorganisms may be more efficient. So there are ways where photobiological production can beat this if you don't do biomass, if you take out the energy at an earlier stage. Biomass also has a problem that it uses large amount of water. So this is a report took as an example. So miscanthus, one of these important grasses, and poplar, very important biomass crops. Depending on where you grow, you use several cubic meters per gigajoule. 1 gigajoule is what a big power plant produces per second. So you would need 63.8 cubic meters per second of water if you want to produce a gigajoule per second of miscanthus. Compare that to a nuclear power plant next to Uppsala. It uses 140 cubic meters per gigajoule of cooling water. But if you look how much water do you need to split to make hydrogen and oxygen by artificial photosynthesis, it's just 0.1 cubic meter. So when it comes to water consumption, artificial photosynthesis is very lean. You can do artificial photosynthesis almost everywhere except in the driest desert. Because 1 gigajoule is a lot of energy, and 1.1 cubic meters per second, that's a relatively small stream. So solar energy is good, but how do we store large amounts of energy? We can store it in many different ways. Pumping water is one way, but the energy density is not very high. And today we are using about 1 terawatt. We're producing 1 terawatt of the hydroelectric power in the world. Theoretical limits is probably around 1 terawatt. So if we want to store even just as little as 1 terawatt, we would have to pump all rivers backwards. All rivers in the world have to be pumped backwards, and this is not going to happen. So pumped water can only work on small scale. Batteries are good, but they don't have. But fuels have about 100 times better energy density, and it's hard to store energy efficiently over a long time. For example, from summer to winter, in rechargeable batteries, they will lose a lot of the energy inside. So fuels are clearly superior if you want to store a large amount of energy, because they have high energy density, and they also last for a long time. You can store it for half a year or even more. And if you look on our energy consumption, this is only 18% is electricity, and 80% is fuel. Most of it made from fossil sources. So even if electricity is a very good energy carrier, and the consumption is expected to increase, fuels are likely to dominate for a long time, maybe for the foreseeable future. So almost all, all renewable energy technologies that people have thought of or realized, they will target either biomass, they lead to biomass production, or electricity production. There's only one technology that does something else than biomass and electricity. And this is solar fuels, direct processes of solar fuels. And in the Swedish Consortium, we have gathered together mainly in Uppsala. We started a long time ago with very few people. Now we're around 80 people. And people working with microbiology or molecular biology and biochemistry, working on organisms that combine photosynthesis and the capacity to make hydrogen in hydrogenase enzymes, but also they can make butanol, and they can be genetically modified to make long chain hydrocarbons. And they're working much on the fundamental metabolic engineering, as well, but also doing experiments on bioreactors. People in the consortium are also working, looking at the molecular mechanisms of photosystem 2. That is a reaction center that converts sunlight into charge separation and oxidizes water to oxygen. And hydrogenase enzyme that converts protons to hydrogen and vice versa. Many of us work with a synthetic system represented by this cartoon, where we coupled photochemical shaw separation, where absorbing light by one molecular unit, and electrons are shuttled to the catalyst and taken from another catalyst, splitting water and reducing protons. So artificial photosynthesis is the only renewable energy technology that is known, that can lead to, that we have invented, that leads to anything else than biomass or electricity. And it's interesting now to see that there's an initiative called Mission Innovation to accelerate clean energy revolution. And this is after the Paris climate meeting in November-December 2015. 22 countries in the world, including the European Commission, decided to double the clean energy research and development investments over 5 years. So all countries should double their efforts, investments in research and development in 5 years. And that's quite a lot. And we see that Japan is one of those countries. And basically all large economic countries are members there. Sweden is also members. The first action was to launch 7 innovation challenges. And they have been formulated, but the details of the programs are not written yet. They're about to be written now. They should be ready by May 2018. And interestingly, one of these 7 innovation challenges is on solar fuels, converting sunlight innovation challenge to discover affordable ways to convert sunlight into storeable solar fuels. And this has been defined by these countries as one of the important goals for the next few years. And we will be interested to see what, there will be a research program within Mission Innovation that focuses on international collaboration. It's also interesting to see that the World Economic Forum listed liquid fuels from sunshine as number 4 among top 10 emerging technologies in 2017. So solar fuels is on the agenda worldwide, and we have to, this is a great inspiration. And but also we who are working in the field of solar fuels have to make sure that we live up to the promises. So, I said that biomass production is very inefficient. That is true. But the primary processes of photosynthesis are very efficient. And in photosystem 2, you can see there's a big protein complex. And you only see here in color the molecules that are directly involved in light-induced shaw separation. So here you see in green chlorophylls, in brown chlorophyll-like molecules called pheophytin, and in purple quinones. And here on this side you see the water oxidizing complex. It's a cluster of 4 manganeses and 1 calcium, and there's a tyrosine involved in the middle between the calcium manganese cluster and the chlorophylls. And when the photosystem 2 is excited by light, 1 electron moves from the chlorophylls in several steps out to the quinone. And you form a highly oxidizing chlorophyll radical. And this chlorophyll radical is the most highly oxidizing chlorophyll in nature. And this is able to oxidize the donor side. So in a proton-coupled electron transfer reaction, the tyrosine Z is oxidized, and this radical is then oxidizing the manganese cluster. So you go from single electron transfer to proton-coupled electron transfer to multi-electron water oxidation on the manganese cluster. So photosystem 2 does a remarkable job, and it has for a long time been a great inspiration for artificial photosynthesis, because these are the primary processes we like to mimic or be inspired by. We don't want to copy the poor efficiency of biomass production, but we'd like to copy these principles in an efficient man-made system. Yes, and this is a more recent structure. We actually see a atomically detailed structure. This is from Shen's work, Nature in 2011. And so the tyrosine radical has given a proton away to the histidine that is nearby. And the question is then whether the proton goes back from the cluster or from this histidine. So the many details here that we don't know exactly what the oxygen-oxygen bond formation step of this cluster. But there's a very intense research going on to finalize the details of this function. What this does though is that this catalyst is able to give 4 electrons away before it completes water oxidation. So photons come in one by one at some time interval. And every time a photon comes in, an electron is sent from the manganese cluster to tyrosine and to chlorophyll. So one flash, one photon, 1 electron goes, and a proton. Second flash, 1 electron. Third, 1 electron and a proton. And then the fourth photon, 1 electron, 2 protons are released, oxygen is released, and you complete the cycle. The Fukushima and Honda paper in 1972 was a great inspiration because for the first time it was shown that a man-made artificial system could do water splitting. This is titanium dioxide with a band gap in the near UV. So they could only use ultraviolet light, and they had to put on a little extra bias, a little extra chemical electrical potential to complete the hydrogen production. But it could show with some efficiency that you could split water into oxygen and hydrogen in a man-made system. Some years later, John Turner made a much more efficient device with an impressive 12.4% energy conversion efficiency. This was a very expensive construct, very expensive semiconductor materials and platinum electrodes. And it also, after a few hours, there were large holes in the electrodes, so it wasn't very stable. But it was a great progress at the time. What we see today are systems that are more stable and also made without expensive semiconductor, or at least without very expensive semiconductor, without precious elements. You can see this is silicon photovoltaics. It's still a bit too expensive construction, but it's not this space technology semiconductors of John Turner. And we see nickel, molybdenum, zinc elements here as the catalysts. This is an example from a Swedish group. They coupled perovskite semiconductor to a nickel cobalt oxide nanorods on carbon paper. So a cheap material catalyst from 6% solid to hydrogen. So the outline of my talk is basically this. This was my solar fuels introduction. And what I will talk about now is that I think molecular catalysts have a potential to be much better than materials. So all the examples I showed you so far were materials-based systems. And they are better today. But I think in the future, molecular catalysts can be much better. And here I want to discuss what we can learn from nature. I will then show you a little bit about my work on proton-coupled electron transfer and why I think this is very important in artificial photosynthesis. Finally, I will go through some of my ideas on mechanistic understanding of catalysts and molecular devices and the work we do there with time-resolved spectroscopy. So synthetic molecular systems, or in particular maybe hybrid systems where you combine semiconductors for charge separation with molecules for catalysis, have a higher potential because you can tune a molecule, the molecular properties, much more than you can tune a material. So traditionally, you could tune a material by changing the composition, changing the phase. I mean, you could be rutile or anatase, for example, for titanium dioxide, and you can change the facet. But that was about it. Then the nano-revolution came, and people got very excited because you could tune the material's properties by the morphology. But think about how we can tune a molecule by its morphology. Small changes of distances and bond lengths and substituents, you can tune the molecular properties enormously. And if you can tune them, I think you can make them work much better. But the more you can tune, the more knobs you can turn, the harder it is to turn them right. So it takes longer time, I think, to find the right solution for a molecule. But eventually, I think it's going to be much better. We can get mechanistic information. This is easier with a molecule, not easier, but easier. And mechanistic information makes it better to do intelligent design of a catalyst. People often complain about stability, that molecules are unstable. And this is often true. But I think it's very important to remember that the Grätzel cell, the dye-sensitized solar cell, that used for 15 years as the champion dye was a ruthenium complex that in solution didn't last an hour in sunlight. It was very unstable. But in the device, it lasted for 20 years. And this shows a very important principle of kinetic stabilization. We think we know why the ruthenium dye is so much more stable in the device. Because the lowest excited state of that ruthenium dye is a metal to ligand charge transfer state. This is the state that does the interesting photochemistry, which is in the device injecting an electron in the titanium dioxide conduction band. But slightly above that state is a metal center state, which is DD star excitation. And this means that you're populating an antibonding orbital along a metal ligand bond. So you have a tendency to dissociate ligands. So this is believed to be the main source of ligand loss and complex decomposition. But if you outcompete that process kinetically by an ultra-fast electron injection, you can stabilize the dye. And I think this is something that is general. If we set up a device to work well, we're going to have rapid forward reactions that are productive, which will outcompete the degrading reactions. So for a well-functioning device, we will see kinetic stabilization a lot. And this means that catalysts, sensitizers, can be much more stable than we expect from their behavior in isolation. Enzymes are great examples of molecular catalysts. And this is a carbon monoxide dehydrogenase from Holger Dobek's work. And here you see a nickel iron center that binds the carbon dioxide. So you see a nickel carbon and an iron oxygen bond, and two hydrogen bonds are drawn here. So there are multiple detailed interactions with the substrate, multiple binding modes. And you see that the carbon dioxide is modeled as being bent. And normally the free carbon dioxide is straight, and this is bent, which means it is much easier to reduce it. So you see in the three-dimensional structure around the substrate, you are fine-tuning the interactions with the substrate. And this is something that is very hard to do in a material. If you take a very small nanoparticle, 3 nanometers in diameter, it has this size, huge in comparison. And it's very hard to do this fine detailed tuning of interactions on that large surface, and especially the three-dimensional arrangement around the carbon dioxide is very hard to do. And there where I think molecules may have an advantage. So there has been a lot of progress on molecular catalysts in recent years. And for a long time, water splitting was thought to be the holy grail of artificial photosynthesis. If you're not familiar with what the holy grail means, it is from the legend of King Arthur, where his knights were supposed to find the holy grail, which was a chalk that Christ was drinking from the night before he died. And the point was not that they should find it, but looking for it. gave them, kept their minds clear and the heart pure. And this was a perfect analogy for a long time for artificial photosynthesis. We had never thought that we would reach there. But if you look at my next graph, this is a collection of molecular water oxidation catalysts that work on the with serum-4 oxidation in acidic medium. For a long, long time, we only had one, Tom Meyer's famous blue dimer, that worked very, very slowly. So turnover frequency of 4 milli turnovers per second. So that means how many molecules of oxygen that are produced per second. And then it took more than 20 years until Tony Joubert made the second catalyst. But then progress has been much quicker, and under these conditions, Li Cheng-Sun in the Swedish Consortium in Stockholm, he has the best catalysts. And they are now up to turnover frequencies of more than 1,000 per second and more than 10 to 5 turnovers. And this makes me optimistic, because if you look back where we were 10, 15 years ago, and if you dare to extrapolate and believe that this development will continue for 10, 15 years, then we will be up to 10 to 8 turnovers intake. And 10 to 8 turnovers is where an industrial catalyst is supposed to be today. This is of course under serum conditions. This needs to also be realized in a functional device. But still, there's reason to be optimistic, but the progress has really, truly been great. And one reason for this progress is that we understand a lot of how this catalyst works. If you take this ligand here, it has a relatively small bite angle, leaving a rather open site here. And this means that water can come in, and you can actually form a heptacoordinate species, 7 coordinate species. And Li Cheng has evidence from several techniques that this is a mechanism that the ruthenium oxygen species. This is the water that has come into ruthenium. When the ruthenium is oxidized, the water is deprotonated, and finally you form an oxygen-oxygen bond between 2 catalysts working together. And Li Cheng has stabilized this structure by increasing the pi-pi stacking between these units. Another enzyme that is interesting to look at is iron-iron hydrogenase. This turns over protons to hydrogen in the best enzymes with a turnover frequency of 10,000 per second. And with very low overall potential. This is a little unusual structure, cyanide and carbonyl ligands, and you have a bridging dithiolate unit. But this can be mimicked. This structure can be mimicked in synthetic complexes, and there are more than 200 complexes of this kind that have been shown to be electrocatalysts. So this is a mimic. You see the dithiolate bridge with the nitrogen bridgehead, carbonyls and cyanides. But this is a pretty lousy catalyst, not very good at all. It works, but compared to nature, no. It has a larger overpotential, and it's rather slow. But then Gustav Berggren, who was a postdoc with Vincent Artero and Mark Fontecave, he's now an assistant professor in Uppsala. He did a crazy experiment. He took an apo-hydrogenase. This is a hydrogenase where you take out the active site, and he just mixed them. And this is Gustav didn't have any molecular biology training, no biochemistry training, and that was good because he wouldn't have dared to do this. And Mark Fontecave strongly advised him to not waste his time with this experiment. But he was of course very happy that Gustav did, because this turned out to work. You take a lousy catalyst, you put it into an apo enzyme, and you get a functional enzyme, functional catalyst, that functions with identical catalytic properties. Spectroscopy tells us this is exactly the same structure as a natural enzyme. So this is not the way nature assembles this complex, absolutely not. But Gustav could make the analog with instead of nitrogen is an oxygen or a carbon in the bridgehead. It goes in, and it still has an oxygen and carbon in the bridgehead, and there's absolutely no catalytic activity. And this tells us, in addition to being very cool biochemical work, this tells us something about design of catalysts. What did we really do? Going from a lousy catalyst to a great catalyst. We modified the third and fourth coordination sphere. Maybe it's not enough to just work on the first coordination sphere to tune the catalyst. Maybe we have to work on the surroundings of the catalyst. But do we have to have an entire protein matrix for the molecule, for the catalyst to be good? The protein matrix here in question, this protein has a molecular weight of 100,000. We want to make smaller molecules than that. But could we fake it a bit? Take the most important parts of this and get similar effects in the synthetic complex. Well, my colleague here, Sasha Alt in Uppsala, he took this ruthenium, made this ruthenium complex, which is a carbon dioxide catalyst. It was already known that if you put terp-butyl groups here on the terp-pyridine ligand, that this increased the rate of carbon dioxide reduction. But what Sasha did was to put a methylene group here in a sterically demanding position to distort the complex. And what he got was a complete change in mechanism. So normally, without the methylene group, you have to reduce it, putting an electron on the terp-pyridine, and reduce it a second time to put a second electron on this ligand. And only then do you have enough electron density that the carbon dioxide can bind, and the electron density moves up in the carbon dioxide, and you continue the catalytic cycle. So electron, electron, and then substrate binding. With this methylene group, you reduce the complex once, and already then the carbon dioxide is able to bind. And then it becomes much easier to reduce it the second time. So electrochemical potential for this step is much milder than that one. So the overall potential of catalysis decreased by 400 millivolt, which is huge, just because this methylene group. So steric interactions is something we can work on, which sort of say mimics what the enzyme does. It does a lot on the sterics of the first coordination sphere. Sasha also worked on immobilization in matter-organic frameworks together with Seth Cohen in San Diego. So they took the matter-organic framework called UIO66, where you have zirconia clusters held together by dicarboxybenzene. And they form three-dimensional networks. This is just one cube and it just continues. It's a polymer, three-dimensional polymer. And builds particles on the hundreds of nanometer size. And what they did for the first time was post-synthetic exchange. So when this matter-organic framework is formed, they soak it together with this catalyst that looks like the linker molecule, but it has also the catalytic unit here. And spectroscopy and diffraction told them that the catalyst and the MOF was intact. And when they run a photochemical experiment, the ruthenium trisbipy as sensitizer with ascorbate, you form the reduced ruthenium trisbipy that reduces the catalyst, and you form a hydrogen. Without the matter-organic framework, catalysis works for a while and then stops. And when you look with the infrared spectroscopy on the carbonyls here, the catalyst is dead. There's no catalyst left. But in the matter-organic framework, catalysis works for much longer. And when you take the infrared spectrum here, the catalyst is intact. It hasn't degraded. So there are other effects on this side that leads to the stop. So this is an increase in stability that is much larger than just comparing these two. I think it's almost a magnitude increase in stability. So why do we stabilize a molecule in a MOF? And the honest answer is, of course, we don't know. We don't have any direct evidence. But the reasonable hypothesis is that the reduced complexes have a tendency to dimerize. When they dimerize, they can lose a carbonyl, and then you leads to a chain of reactions where they probably polymerize and precipitate. When you immobilize them, you of course hinder dimerization. So in principle, any kind of simple immobilization could be an advantage for stability on molecular catalysts. So this is a very simple function of what the enzyme does. The enzyme covers the active site and protects it from other active sites. Controlling proton-coupled electron transfer. Enzymes are very good at doing this. Acid-base group near the metal center may accelerate catalysis by helping it to remove or add protons. The famous, this is very popular right now, and the most famous example is from Dan Du Bois and his co-workers in Pacific Northwest National Lab. The nickel P2N2 to phosphine and 2 nitrogen amine ligands, amine functions in the ligand. This coordinates with the phosphines, but the amines are there as proton shuttles. And what they believe happens is that the amine is protonated and then shuttles the proton to the nickel, forming a nickel hydride. And this is being discussed as having a base nearby. But my point is that having a base nearby is a very, very loose concept. What does it mean having a base nearby? I will show you examples from my work where you have to where you can show that you can have a huge effect by having a base nearby, or sometimes a small effect, and you have to control the position of that base very, very well. And this is proton-coupled electron transfer. So I've been working for a long time inspired by, this is where we started actually with proton-coupled electron transfer. We wanted to make a mimic of the donor side of photosystem 2. Chlorophyll, tyrosine, histidine, which is near the manganese cluster. So we took ruthenium instead of chlorophyll. We give it a nanosecond laser flash and send out an electron to an acceptor in solution. Then we have photo-generated a strong oxidant that is able to oxidize the tyrosine residue here. Then we can have different bases so that can accept the proton, or the proton goes to the water. And we can follow these processes by transient absorption changes. We can see the radical being formed, and we can see the ruthenium being reduced again. Concerted proton-coupled electron transfer. This is for those of you who are familiar with Marcus theory. This is just one slide. But this is a reactant state, this is a product state, and just like in normal electron transfer theory, the solvent need to reorganize because electrons and protons are charged. So when they move, the solvent dipoles are reorganizing. And you have to reorganize to find a common crossing point here where the electron can tunnel. But in proton-coupled electron transfer, and then you evolve further on the product state. But in proton-coupled electron transfer, you also have a proton tunneling. Protons are much more heavy than the electrons, so they have very localized wave functions. So if this is the reactant state and this is a product state, you see that they can become isoenergetic at the transition state. And the wave function for the donor and the acceptor state are in blue and red. And the overlap between these wave functions, these tails here, is what determines the probability of proton transfer. And this was a series of compounds we made. This was the first series of model systems that probe the distance dependence of conservative PCT. So here we took a tyrosine with either a benzimidazole or a pyridine, and then we linked it directly or via methylene group. And thereby we varied the distance between the oxygen and the nitrogen between 2.55 and 2.75. Bearing in mind that each bond is about 1 angstrom, it means that we vary the distance the proton has to tunnel from about 0.55 to about 0.75 angstrom, on the order of 0.6 angstrom. So the proton, if this is the oxygen, This is a nitrogen. My thumb is the proton. The proton has a tunnel from here to there. And then the distance here is changed. And if the wave function is localized, 0.2 angstrom difference here makes a huge effect on the overlap. And you can see how large the effect is. This is the exponential. This shows an exponential dependence of the rate constant for proton-coupled electron transfer on the distance. And the slope of this exponential dependence is 27 per angstrom. For those who are familiar with electron transfer, this is a lot higher. For electron transfer, it's about 1 per angstrom, roughly. And 1 per angstrom means that an electron can tunnel 10 angstroms, 20 angstroms, no problem. But 27 per angstrom is a dramatic distance dependence. So with only 0.2 angstrom difference, we change the rate by a factor of 100. I think this is very important for catalysts. Catalysts work often with rate-determining PCET steps. And we may have spent too much time worrying about the electron transfer part and too little time worrying about the proton part. Because this is much more sensitive. And this is why I mean that having a base nearby is a bit too imprecise. This is nearby, this is nearby, this is also nearby. 4 angstroms is also nearby, but that's a very, very long tunneling distance. So you have to be very careful here. If you have a catalyst that works here, what if you could change the distance slightly and get a factor of 100 faster catalysis? You would like to do that. I skipped that, but okay. We wanted to see, this is for tyrosine. A very, very polar OH bond forms strong hydrogen bonds. What about metal systems then? Metal hydrides are critical intermediates, key intermediates for CO2 reduction and proton reduction or hydrogen oxidation. But typically, people discuss the mechanism here, now I take an iron-iron system, in terms of square schemes. So one electron addition goes down, one proton addition goes to the right. So people discuss mechanism in stepwise electrons and proton transfers. But what about the concerted reaction, where you're actually transferring both at the same time? And the concerted reaction had never been demonstrated for a metal hydride before. Note that I'm not talking about hydrogen atom transfer. That's common, where a metal hydride is transferring a hydrogen atom to organic radical, for example. But in solar fuels, we don't have hydrogen atom transfer because the electron acceptor and the proton acceptor are not the same. This is a Shaw separation system, and this is a base. They are separate. So it's a bidirectional proton-coupled electron transfer. They go in different directions. So I wanted to see if you could get a concerted reaction from metal hydride, because I believe that the concerted reaction may give a lower barrier. And the reason for that, and a lower barrier is great, because that is what you want. That means an energy efficient reaction, low overpotential catalysis. And in this paper, we could show this. We could show a concerted reaction. So we took this tungsten hydride as a model of a metal hydride, model of a catalytic intermediate. And we were oxidizing with metal BIP oxidants and pyridine bases with a range of different pyridines and oxidants. And we could show that if the oxidant is weak and the base is weak, it prefers to go this way. And weak oxidant and weak base means an energy efficient conditions. So we think this is important for catalysis. This is how we should design a catalyst. But admittedly, this particular reaction wasn't very fast. So we wanted to see how could we speed that up without wasting more free energy. So another very good postdoc, Tianfei Liu, very excellent synthetic skills. He made a series of compounds looking like this with different pyridines, I just showed one, to increase the proton transfer probability. And we could show that we could have a hydrogen bond here. And when he did the similar kinetics now with an intramolecular base, he could get up to more than 10,000 times acceleration of the rate. So 10,000 times is a lot. And it was a first demonstration, direct kinetic demonstration of the PCET step of a catalyst model compound. And we also get a lot of detailed information on the mechanism here, so we can probe. And actually, in this system, by changing the oxidants and the base, we could make it go this way, or that way for the same compound. So that was a short excursion into PCET. I will finish by giving you some information on what we're doing on the mechanistic understanding of real catalysts and molecular devices. So often catalysts are depicted like this. They turn over a substrate into fuel and without being consumed, but this hides all mechanistic information. And if you want to make a rational design of a catalyst, you need to rationally evaluate and redesign and compare different catalysts. And this is a problem because turnover frequencies and turnover numbers that we often use, they are strongly dependent on conditions. And this means that if you and I compare a catalyst and you do under some condition and I do under other conditions, it's hard to say which one is best. And conditions in future devices is not known. No one knows what artificial photosynthesis will look like eventually. So some people want us to benchmark so that we agree on this is one set of conditions everyone should use. But what if these conditions are completely rubbish? What if they are not at all what we're going to use in the device? What's the point of the benchmarking on those conditions? I think the solution is we need mechanistic understanding so we can predict what a catalyst, how a catalyst will work if you change the conditions. And one thing we did was using nanosecond infrared spectroscopy. So basically it's a laser flash photolysis experiment where we give a laser flash to a ruthenium compound in the presence of a donor, and on a nanosecond time scale the reduced ruthenium does a single electron transfer to the catalyst. So you can control single electron transfer. And for this particular compound in electrochemistry, it has inverted potentials to immediately go over there, and then catalysis works this way. But and you can not get any information on these short-lived intermediates. But in our transient absorption spectroscopy on the nano to microsecond time scale, we can see the reduced catalyst being formed, and we can see it being protonated. And we can get information on the structural changes that occur, and on the kinetics. And this shows also that photochemical and electrochemical catalysis follows different mechanisms, which also highlights the problem with benchmarking. What if you benchmark electrochemically and then you're going to make a device running under photochemical conditions? Or vice versa. We also did time-resolved infrared spectroscopy to look at this system. This was published by Durant, Perus, and I²CNER a couple of years ago. They took basically the famous rhenium tris-carbonyl compound, but linked it via phosphonate group on titanium dioxide. This is famous for doing carbon dioxide reduction. So with visible lights, in DMF solvent with triethanolamine as a sacrificial donor, they could show carbon dioxide reduction, but when they put it on titanium dioxide colloids, it worked much better. But now we wanted to understand this mechanism, and my colleague, an associate professor, asked me, "What? Are you crazy? Why do you put it on titanium dioxide? Because we want to do reduction. We want to do reductive chemistry, and titanium dioxide is known for taking electrons. Why would you want to put it on titanium dioxide?" So I explained, okay, no, but they said that there is no electron injection in this. This just works as a scaffold. There's no, and it improves the catalysis by preventing dimer formations. But we found that this is not the case. First of all, we found, like Ishitani sensei has already shown, that in DMF with triethylamine, you form and carbon dioxide, you form an adduct, carbonate adduct. So carbon dioxide and triethylamine binds as a ligand to rhenium. And on the femtosecond, picosecond timescale, we have electron injection, also from this structure, into titanium dioxide. But immediately, the whole is shifted out of the triethylamine, because it's coordinated. And here is the improvement in efficiency thanks to the titanium dioxide. Then, surprisingly, we found that on the nanosecond timescale, a second electron comes from the radical, because this radical you form here is very, very reducing. So, in about 1 microsecond, we have gone from the oxidized complex to the neutral complex to the reduced complex. And this we know from the transient infrared. This is a nanosecond laser flash photolysis experiment again, where we have infrared lasers to look at the products. And this is if you do the experiment with triethylamine on zirconia, zirconia is inert. Here you can't have any electron injection, and we don't see any reduced rhenium. So triethylamine is not very efficient in reducing the excited state. If you do the experiment with the titanium dioxide without triethylamine, we get the oxidized rhenium. There is nothing that can take the whole, so we get the oxidized rhenium, and it has a band over here. And we also see conduction band electrons that lift the baseline. So the entire infrared baseline goes up because of conduction band electrons. So electrons are injected indeed into titanium dioxide, but we see oxidized rhenium. But with triethylamine, we see electrons in the conduction band, and we see reduced rhenium. So this electrons in conduction band, reduced rhenium. And the only thing that can carry the holes is the triethylamine. Then we see a very slow disappearance of these conduction band electrons at the same time as the reduced rhenium disappears. So we propose that you actually have that this electron goes back to the complex where CO2 has bound in the right way for catalysis. So we actually go into the continue the catalytic cycle. So this is invest 1 electron into titanium dioxide and get 3 electrons back. That's not good. That's not bad. That's a great bank deal. I wish my bank worked like that. And the reason that titanium dioxide works much better than just the solution is that this injection into titanium dioxide gives a long-lived and strongly oxidizing rhenium, which is very efficient in oxidizing the triethylamine that is already assembled to the metal complex. So I will just very briefly show that we're working also on towards tandem cells. Tandem cells where you have a photoanode and a photocathode. So photoanode is typically titanium dioxide that is shown. You have a dye-sensitive, like in a dye-sensitized solar cell, you excite the dye and inject an electron, and the oxidized dye oxidizes the catalyst for water oxidation. On the other side, you have another semiconductor, typically nickel oxide, where the excitation of the dye leads to electron transfer from the valence band, or sometimes we call it hole injection into nickel oxide. And the reduced catalyst dye gives an electron to the catalyst that does hydrogen production. This side works better than this side, so we have decided for a long time to try to work on this side. And we have made several dye-sensitized solar cells, but these are our first results, including a catalyst. So we wanted to bind a dye, a coumarin dye, and an iron catalyst side by side. So we would not have to link them together synthetically, because it makes life simpler. And hole injection worked. So on the 100 femtosecond time scale, whole injection has converted the excited coumarin into the coumarin radical. Then if you look on longer time scale, the coumarin radical in purple here has gone to a different species here with a half-life of about 10 picosecond. And this can be identified as a reduced catalyst. This is a reference measurement. And the reduced catalyst survives for over 1 nanosecond. And if you look at the reduced catalyst on long time scale, you see it disappears on the microsecond time scale. So this shows that you don't form isolated islands. The coumarin is able to react, all coumarin is able to react and give an electron to the catalyst. And there is a relatively slow recombination between the reduced catalyst and the nickel oxide holes. So the question is, can we continue with protonating this and do photocatalysis? To do this, we change the linker because now we want to be in water, so there's a different linker, but essentially we see the same kinetics here and even slightly faster electron transfer. But here we could get the first evidence for, by transient infrared of the catalyst. So here, without the catalyst, just the coumarin and nickel oxide, we see a general temporarily broad signal related to the nickel oxide. But when we have the catalyst there, we see the carbonyl features here. The carbonyl bands that shift. This shifts from here to there, and here to there, here to there. So, and the rise of that shift is about 3 to 4 picosecond. Then we have some decay, and the rest of it is stable. So 3 to 4 picosecond agrees pretty well with what we see in the visible. We made a device. When we turn on light, there is a photocurrent. When we turn off, it goes away, and we can continue for a while. Hydrogen is being produced. So it shows that we can get a cold-sensitized photocathode, higher photocurrent with the catalyst. We get a Faraday efficiency of about 50%, which is pretty good for these devices. There are some losses in the detection methods. And we could also unfortunately then of course the device performance goes down, and we could detect by infrared spectroscopy that before the sample you see strong catalyst bands, but after, when we stop here, about 2/3 have been lost, while the dye is more intact. So catalyst degradation is the main reason for decreased photocurrent. And these were the first mechanistic studies of dye-sensitized photocathodes at all. And it was the first to prove that the catalyst actually binds to the nickel oxide in the first place. So this is maybe not too impressive, but I like to draw the analogy with the first transistor for Bell Labs. It doesn't look very good, but it changed the world. Of course, my hope is that our devices and the people that our colleagues develop will transform the world as much as the first transistor did. It will take a little while, but hopefully we'll get there. So conclusions from this part is that transient infrared on many time scales can follow and identify catalysts and intermediates and get kinetic information. This is some femtosecond infrared. We can get dye-catalyst surface electron transfer with high rates and efficiency. And we made the first molecular photocathode solar fuel device with proven catalyst binding and mechanistic information on catalyst electron transfer. So this was to remind you of the summary. I discussed motivated why solar fuels, why I believe that molecular catalysts can have the potential to be much better than materials, and what we can learn from nature to incorporate in the design. Told you about the importance of proton-coupled electron transfer and some of our work on mechanistic understanding of catalysts and molecular devices. And with that, I'd like to thank the people who were involved, the people who gave us funding, and the entire Swedish Consortium for Artificial Photosynthesis. And I'm happy to answer any questions.